Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is an event of the International Society for Extracellular Vesicles, and my name is Kenneth Witwer. I am the chair for Science and Meetings for ISEV, and this week we're having a special collaboration with SOCRATES, which is the Singapore Society for Extracellular Vesicle Research. And we were just discussing before the event that this is our eighth session that is in collaboration. So thanks very much to Wei Seong Toh, to Minh, who have made this happen, along with the many active extracellular vesicle researchers in Singapore. And so we're going to have another very nice presentation today. And before we get started, I would just like to remind everybody that you can place your comments and questions in the chat box, and then we will unmute you at the end so that you can interact with our presenter. So I'd like to turn this over now to Wei Seong Toh, who is joining us from San Francisco for a change at the International Society for Cell and Gene Therapy meeting, which I will also be attending starting tomorrow. And I've heard there are a lot of good extracellular vesicle presentations there. So Wei Seong, thanks for helping to host and moderate today, and I'll leave it to you now.
Thank you, Ken. So this is Wei Seong Toh from National University of Singapore, and it's my great pleasure to introduce Dr. Jia Tong Loh from A-Star Singapore. Dr. Loh is currently a Senior Research Fellow and a SIGN Fellow under the supervision of Professor Lam Kong Peng at the Singapore Immunology Network in A-Star. Her research interest lies in deciphering the innate immune signaling pathways and further translating this knowledge for the development of potential therapeutics against immune-mediated diseases. Today, she will share with us her latest collaborative research investigating the mechanism of action of MSC-sEV in the attenuation of neutrophil and complement hyperactivity. Over to you, Jia Tong.
Thank you, Wei Seong, for the introduction. So let me share my screen first. So firstly, I would like to thank Socrates and the EV Club for their invitation to this seminar series. So I'm really excited to be here today to share with you our recently published work. So I have an immunology background, so I'm not an expert in the field of extracellular vesicles. So I would like to first acknowledge Dr. Lim Sai Kiang for initiating this work with us, so we could venture into a new and exciting research area, looking at the therapeutic potential of EVs and how they interact with and modulate the immune system. So today I'll be sharing with you our work done in collaboration with Dr. Lim Sai Kiang's group, which investigates how MSC-sEVs attenuate the crosstalk between neutrophils and the complement system. So as we all know, the COVID-19 pandemic has affected more than 470 million individuals and caused over 6 million deaths to date. Although majority of these patients are presented with asymptomatic or mild disease, more severe and critical illness can still arise in a subset of these patients because of the dysregulation of their innate immune response. And this leads to the development of cytokine storm, ARDS, multiple organ failure, and even death. So more importantly, breakthrough infection is increasingly common in these fully vaccinated individuals because of the emergence of new SARS-CoV-2 variants. Therefore, it is necessary for us to expand and diversify our therapeutic toolbox against SARS-CoV-2 in order to counteract this ongoing pandemic. So one immunological indicator of poor disease outcome in COVID-19 is the increased numbers of circulating neutrophils. So we can see that there is higher number of neutrophils in the severe COVID-19 patients as compared to the healthy and mild and moderate patients. So these neutrophils, they will actually migrate to the infected pulmonary sites where they undergo degranulation to release massive amount of cytokines, pro-inflammatory mediators such as calprotectin, as well as reactive oxygen species. And all this together can actually trigger tissue damage. And in addition, the activated neutrophils will also release net-like structure called the neutrophil extracellular traps, which basically are actually composed of granule proteins, DNA, and chromatin that are extruded from these neutrophils as an attempt for them to trap the invading viral pathogen. But however, the sustained formation of nets can actually predispose the patients to thrombosis, organ damage, and even death. So given that neutrophil-associated inflammation plays a critical pathogenic role in ARDS, therapeutic strategies that actually targets these hyperactivated neutrophils have been proposed as potential therapeutic strategies to treat severe COVID-19. So apart from neutrophils, the complement is another arm of the innate immune system that is strongly associated with the clinical severity of COVID-19. So the complements can help to eliminate the viral pathogens directly and indirectly through opsonization, formation of the membrane attack complex C5b9, as well as the recruitment of other leukocytes to promote an antiviral inflammatory response. However, clinical data have actually revealed that the increased activation of complement system is a distinct feature that correlates with worse COVID-19 outcomes. So we can see that elevated levels of circulating complement markers such as C5b9 were actually present in patients with COVID-19 as compared to other etiologies of acute respiratory failure such as influenza. And these markers can actually distinguish those with worse outcomes. And this is because the overactivation of the complement system can actually contribute to cytokine storm, endothelial inflammation, and hypercoagulation, which then leads to the organ damage. Therefore, various complement inhibitors are actually being explored in multiple phase 2 and phase 3 clinical trials to combat the SARS-CoV-2 infection. So as you all know, mesenchymal stem cells and their EVs are known for their immunomodulatory and regenerative properties, and therefore they are also proposed as potential therapies for severe COVID-19 patients. And indeed, several preclinical and clinical studies have actually demonstrated that their safety and efficacy in mitigating the symptoms that are associated with severe COVID-19. However, the precise mechanism by which these EVs exert their effects to benefit the COVID-19 patients still remains unclear. So this led us to the aim of our study, which is firstly to investigate whether the complement system and neutrophils, the two key players involved in COVID-19 pathogenesis, whether they are interrelated components of a broader immune cascade. So specifically, we want to understand if the terminal complement activation complex, C5b9, can directly drive neutrophilic immune responses. And if yes, whether MSC-sEVs can actually modulate this immune axis and also the mechanism by which this modulation occurs. So firstly, to characterize the effects of complement activation on neutrophil response, we purified neutrophils from blood of healthy human donors, and we initiated the complement activation by adding C5b6 and C7 followed by C8 and C9. So these complement components will assemble sequentially into this ring-shaped structure, which then inserts deeply into the membrane lipid bilayer to form a small leaky pore. So let us confirm the assembly of this terminal complement complex C5b9 on neutrophil surface by flow cytometry. So we can see by the sheaf in this histogram that there is a successful assembly of C5b9 on the neutrophils as compared to the control. And here we observe that C5b9 assembly actually triggers release of nets by neutrophils, as we can see from the citrulinated histone 3 staining, which is a distinctive marker for nets formation, as well as this formation of this extracellular network of DNA fibers. And we can see from our quantification of the DNA size, which tells us whether netosis occurs or not, that over 60% of these neutrophils actually undergo netosis upon C5b9 assembly, and the area of the nets were actually significantly larger than that of the control group. Therefore, assembly of C5b9 on the neutrophil surface actually promotes netosis. So we also want to look into other neutrophil-associated inflammatory mediators associated with COVID-19 severity, such as IL-17 production, as well as the calprotectin S100A8 and A9 levels. So according to our RT-qPCR and ELISA analysis, we can see that there is actually a significant induction in IL-17 at the transcript level, as well as the protein level, which is the secretion into the culture medium upon C5b9 assembly. But on the other hand, the transcript level of S100A8 and A9 actually remained unchanged. So taken together, this data actually tells us that hyperactivation of the complement system and neutrophilic immune response in these severe COVID-19 patients they are likely to be interrelated components of a broader immune cascade. So this feed-forward loop between C5b9 and neutrophils can then escalate inflammation, tissue injury, and thrombosis that ultimately leads to respiratory failure, as well as other devastating complications which are observed in these severely ill COVID-19 patients. So MSC-EVs, with their well-documented immunomodulatory properties, have been proposed as therapeutic options for COVID-19 because it can help to alleviate complications such as ARDS. However, the mechanism by which they exert their effects still remains unclear. So we wanted to test whether the MSC-EVs can intervene in this complement-induced neutrophilic immune response. So we assembled C5b9 in the presence of EVs. So here we can see that C5b9 induced the release of NETs by neutrophils, as seen from the citrullinated histone 3 staining, as well as this network of extracellular DNA fibers. And the addition of EVs can actually abrogate NET release. And a concentration as low as 1 microgram per ml is actually sufficient for inhibition. So since complement activation can also trigger IL-17 production by neutrophils, we also investigated the effects of MSC-EVs on this process. So similarly, we see that C5b9 assembly actually induces IL-17 secretion as well as production by the neutrophils at transcript and protein levels. And the addition of EVs is able to attenuate this production of IL-17 in a dose-dependent manner. And we observe that maximum inhibition is reached at approximately 10 microgram per ml. So altogether, this results actually shows that MSC-EVs can attenuate C5b9 mediated NET formation as well as IL-17 production by the neutrophils. So apart from C5b9 assembly, several other stimuli are also known to trigger NETs formation and IL-17 release by neutrophils. So we wanted to determine whether the inhibition of NETosis and IL-17 by these EVs are actually specific to complement-mediated activation of neutrophils. So we went to test this by treating neutrophils with another stimuli called PMA. So PMA can activate neutrophils through the signal transduction enzyme called protein kinase C. And in it, we can see that PMA treatment induced NET release by neutrophils, as seen from this massive formation of extracellular DNA fibers that is positive for his citrullinated histone 3. But however, in this case, we observed that the addition of MSC-EVs at increasing concentration did not have any effect on NETosis. So we also look at IL-17 production, and we observe a similar trend. So we can see that PMA induced IL-17 production and secretion by neutrophils, but the addition of MSC-EVs did not affect the IL-17 levels. Therefore, the MSC-EVs can actually suppress complement but not PMA-mediated activation of neutrophils. Therefore, this tells us that it is likely that the EVs actually exert their inhibition on the complement cascade itself and not through the neutrophil per se. So we know that the assembly of the terminal complement complex C5b9 needs to be a tightly regulated process because we need to protect the host cell from cytolytic complement attack. So the assembly of C5b9 can be inhibited by CD59, which is a widely expressed GPI-anchored membrane protein that is expressed on various cell types, including red blood cells and endothelial cells and leukocytes. So CD59 can exert its effect by binding tightly to the C5b8 complex, and this will inhibit the further incorporation of C9 molecules into this complex and thereby block the formation of this large lytic pore. So previously, Dr. Lim's group actually reported that CD59 is also present on the MSC-EV surface. So they observed that the complement-mediated lysis of sheep red blood cells can be inhibited by MSC-EVs in a CD59-dependent manner. So they can see the addition of exosomes actually attenuate the effect, and the addition of anti-CD59 antibody actually abrogates this inhibitory effect. So this suggests that MSC-EVs can inhibit the assembly of the terminal complement complex through the expression of CD59. So here we wanted to test whether CD59 on the EVs is also required for the suppression of this complement-induced neutrophil activation. So we added a neutralizing antibody against CD59 together with the mixture of the activated complements and neutrophils. So C5b9 addition actually induced netosis in neutrophils, and the EVs can attenuate this effect as mentioned previously. But strikingly, when we add anti-CD59, we observe that it actually abrogates the inhibitory effects of the EV on complement-induced netosis. So the nets released by neutrophils in the presence of EVs and anti-CD59 were actually comparable signs to those found in the control group without any EV treatment. So this suggests that CD59 on MSC-EVs can inhibit C5b9 formation, and this will then abolish the complement-mediated neutrophil netosis. So we further confirm this inhibitory effect of MSC-EVs on IL-17 production, and we can see that inhibition of IL-17 released by EVs was also abolished by anti-CD59 treatment. As such, we have actually identified CD59 to be the MSC-EV attribute that mediates their activity in this case. So CD59 on the EVs can inhibit formation of the C5b9 complex on neutrophil surface, and this will attenuate their activation in the form of net release and IL-17 production. So in conclusion, our study has actually elucidated this feed-forward loop between two key immunological indicators of severe COVID-19, which is namely the complements and the neutrophils. So initiation of this complement activation cascade can induce net release and IL-17 production by neutrophils, and this process can be suppressed by EVs in a CD55 dependent manner. So collectively, this findings actually provides a mechanistic basis for how EVs can inhibit complement-induced neutrophil activation. Thereby, this provides a scientific rationale for the use of MSC-EVs in alleviating the severe manifestations of these critically ill COVID-19 patients. So this presented work has now been published in Cytotherapy. And if you're interested to know more, please take a look at our paper. And lastly, I would like to mention that this complement neutrophil axis functions not only in the context of COVID-19, but it is also likely to be involved in the pathogenesis of psoriatic patients. So psoriasis is a chronic inflammatory skin disorder that is driven predominantly by the IL-23 and IL-17 pathway. So a recent study by Dr. Lim Sai Kiang's group, which make use of this imiquimod-induced psoriatic model, actually shows that topical application of MSC-EVs can reduce the level of IL-17 and C5b9 in the imiquimod-treated skin. And interestingly, they observed that the EVs, when applied topically, were actually unable to reach the underlying dermis, where most of the immune cells that are involved in the pathogenesis of psoriasis are located. But instead, they are confined to the stratum corneum layer, suggesting that their primary site of action must lie within this region. So although stratum corneum is the outermost layer of the epidermis that consists of dead keratinocytes, literature has actually reported that C5b9 can be deposited at this area, as well as activated neutrophils can also infiltrate into the stratum corneum, where collectively they actually helps to mediate this skin inflammation. So based on all these observations, we believe that the feed-forward loop between C5b9 and neutrophils also exists in psoriatic skin, where C5b9 assembly on neutrophils that are present in the stratum corneum will trigger their netosis and IL-17 release, and this will fuel the skin inflammation. And subsequently, when they apply the EVs topically, they actually can inhibit this assembly of C5b9 on the neutrophils, and this will therefore alleviate this complement-mediated neutrophil activation through the expression of CD59. So this ultimately results in the reduction in IL-17 as well as C5b9 levels in the skin of this imiquimod-treated mice upon EV application. So this data actually support the use of MSC-EVs for the treatment of psoriasis. And I'd like to congratulate Dr. Lim for their successful phase 1 clinical trial of the topical application of the EV ointment for psoriasis. And there is no adverse effect observed among the participants. So hopefully we will be able to see a new generation of immunomodulatory drugs for the management of psoriasis in the near future. So with that, I've come to the end of my talk. So I'd like to thank the following who have helped to make this work possible. So firstly, I would like to thank my supervisor, Prof. Lam, for the many opportunities he had given me during my postdoc journey, and also my RO, Joey, for their help in the experiments. I would also like to thank Dr. Lim Sai Kiang and her postdoc, Dr. Zhang Ping, for conceptualizing this project and their insightful discussions in our work. I would also like to acknowledge Dr. Andre Chu for producing the MSC-EVs and Dr. Lai Runchai for characterization of these EVs. So this work is funded by IAF, PP, and A-Star core grants. So lastly, I'd like to thank everyone for your kind attention. Thank you.
Thank you, Jia Tong. So very nice presentation. So let's look at the chat box and we will try to address the questions that were raised.
Okay,
so the first question from Alan. Why do you think the endogenous complement inhibitory mechanisms are not working?
I guess you mean that why when we add EVs, when in the absence of EVs, C5b9 will trigger this activation of NETosis and IL-17, but when we add EVs, it can attenuate. But why is the CD59 not inhibiting these processes endogenously? So I guess there are actually many factors governing the regulatory pathway. So it actually depends on the stimulus as well as because the assembly of C5b9 on cells is actually a protective mechanism for the host so that it can lyse the viral infected cells so that these cells will not survive and the viral cannot replicate themselves. So I believe it's a kind of survival mechanism for the cells not to inhibit this C5b9 mediated effect on the cells, but addition of the EVs can actually modulate this response. Yeah, so you get what I mean?
Yeah.
It's actually I believe it's a survival mechanism that upon the assembly of this C5b9, the cells have to undergo lysis through NETosis in order to stop viral replicating. And we actually did this in an in vitro system where we purify neutrophils alone. So I guess if it's in a host, there are actually many different modulatory mechanisms, but it actually is not present in this in vitro system. Yeah, because interaction, crosstalk among the different cells can help to suppress some of these activating processes. But because we are bringing it in vitro, the assembly of C5b9 will trigger all this process.
Yes, so I thought so too, because this is an in vitro system that you are able to actually control the interaction and to actually decipher the effect on the neutrophils. So maybe I will also follow up with the question that Alan also asked. Do you see any role of poprotein in your NETosis triggering? And do you actually look into other factors such as CD55 and even factor H?
No, actually, we didn't look into that at all. So I cannot answer your question. Yeah, but it will be interesting to look into it in future as well. So I believe there are a lot of different mechanisms governing this process.
Yeah. Okay. Yeah. So moving on. So another question from Tang Pik Tong. Yeah. Is neutrophil has a short half-life time in vitro experiment. So which time point do you think which time point do you collect the samples after stimulation to perform both the qPCR and ELISA?
Yes, we know that neutrophils upon stimulation will actually die very quickly. So we actually did around 3 hours to 6 hours and we actually tested. So within this time, it's actually the neutrophils will still survive, but more than that, like overnight culture, most of the neutrophils will be dead. So we limit our experiment to around 3 to 6 hours.
3 to 6 hours.
To ensure that the neutrophils are still alive.
Yep. So yeah, so any other questions from anyone? Yeah, so maybe I will also have another question that I have in mind. Is there any plan to kind of like test your these in vitro results, okay, in an in vivo animal model?
Because we are looking it into the context of SARS-CoV-2, so it is a bit difficult because SARS-CoV-2 they have actually host tropism. They infect humans, but not as good in mice. So you have to use this humanized ACE2 mice in order for this infection to work, and it's actually quite difficult to get these mice, as well as the BSL level is also, you need certain containment to work with it. But for the psoriatic part, it will be easier. Like Dr. Lim's group, they have actually do it in vivo to see that these EVs actually can attenuate this crosstalk between complements as well as neutrophils. So they actually did this using animal model to see whether these EVs can actually abrogate this immune axis.
Yeah,
so I believe, yes.
Yep. So thanks. Yeah. So Minh have a question.
Yes. Thanks, Jia Tong. It's an excellent talk, and I really like this work. We actually work on COVID-19 in our lab, and we do have the model as well. I'm very curious to ask if, I mean, from the literature, but maybe that you have read when you started this work, how does SARS-CoV-2 elevate the complements in COVID-19 patients? This is mainly in the circulation, right? Not in the lung or so.
So for these questions, according to the study, they actually realized that it's just a trend that they observed that the complement system are actually much highly increased in patients with SARS-CoV-2 infection as compared to various other infection of the lungs like influenza. But I think it is still not very clear how SARS-CoV-2 is specific in the sense that it can trigger complement only in this form of infection. And yes, this happens in the circulation because they actually sample the blood from these patients to see their levels of different complements in the blood after infection.
Okay, so putting this into the context of COVID-19, how you use the MSC-EVs to treat the patients since the effect is in the circulation when you use an IV injection, and as I understand, MSC-EVs when it go into the circulation, it accumulate in the liver, and so some of them may still act in the circulation. They don't last very long. How much do you need? How much EVs do you need in order to neutralize this effect of SARS-CoV-2? Would it be quick enough, and is it affordable for COVID-19?
Okay, so firstly, I'll say I'm not an expert in EVs, but I'll try to answer your question with what I know. So I was thinking if it is possible to apply the EVs intranasally to these patients, because when it reaches the lungs, that is where the site of most of this inflammation is going on. Even though the complements are elevated in the circulation, they will actually reach the pulmonary site, and the neutrophils will also extravasate into the lungs because there are presence of many virus there. So it is in the lungs predominantly that the NETosis, IL-17 release are occurring, and this will cause the acute respiratory defect. So I believe that other than IV, maybe they can apply the exosomes intranasally, but I'm not sure whether currently there have been any trials to do this. Yeah. And how much EVs should be provided? According to our study, it's not too much. Yeah, because
in the literature there are some study on using MSC-EVs to treat COVID-19, right? Do you know how much do they use and how do they treat?
I'm not sure about that. Yeah. But our in vitro data shows that they actually exert their effect at quite low concentration, like for the event of NETosis.
Okay,
Like,
so
1 microgram is sufficient.
yeah, so I see another question from again is difficult for me to pronounce the name. All right, so congratulations on approaching the clinical trial for treating cirrhosis. Okay, so my question is how do you think topical application of EVs will help in treating cirrhosis, which is a systemic condition, and what is the level of severity of the treated conditions? Any
That's my
idea?
mother from Malaysia.
So psoriasis, it usually manifests itself in the skin, and it's actually caused by the IL-23 released by the skin dendritic cells, which then triggers IL-17 released by the gamma delta T cells in the skin, and this will trigger inflammation in the skin. So by applying these EVs topically to the skin, it's actually like trying to inhibit the IL-23, IL-17 axis, and this can help to limit the skin inflammation. And the level of severity of the treated conditions, I think for Dr. Lim's clinical trial is the phase 1, so it's with healthy donors. So they haven't tested the efficacy on the psoriatic patients.
Yeah. So I suppose although the psoriasis is a systemic condition, okay, but again, it's actually affecting on the skin, resulting in the skin inflammation. So I think the topical application of the EV in this case, it will definitely kind of like help to attenuate any form of inflammation on the skin as a local application. So I have another question. Apart from testing this complement assembly on your neutrophils, have you actually tested on other immune cells?
No, we haven't tested it on other immune cells, but we believe we can see something interesting as well.
Yeah. So to my understanding is that IL-17, apart from producing from neutrophils, are also produced by the Th17 helper cells, right?
Yes, T cells.
Yeah. So do you think there is any form of interaction? I mean, of this. I mean, if we are to test this complement assembly on the Th17 cells, do you think there will be some form of effect?
It actually depends because only certain processes that we analyzed in the neutrophils are actually affected, like NETosis and IL-17 production, but the calprotectin like S100A and A9 expression are not affected. And for T cells, even though they produce IL-17, the pathway through which this is regulated is like totally different from neutrophils.
Yeah.
So we wouldn't really know whether when we assemble complements on the T cells, it will affect, because T cells, they can polarize into Th1, Th2, Th17. So we don't know the addition of EVs, they will affect the polarization to which kind, whether there is a bias towards certain T cell subtypes.
Yeah. So certainly it will be interesting to look into the effect of MSC-EV on the polarization of the on the T cells. Yep. Yeah. So there is another. Okay. So is there any other questions from anyone?
All right. Well, I guess we've come to the end of this session then for today. And I'd like to thank everybody for joining and for all of the excellent questions. Let's also like to thank Wei Seong Toh and Minh for helping with organizing and moderating this session. And most of all, Jia Tong Loh, thank you so much for sharing your research, and we wish you the very best as you continue it. So thank you, everybody. We hope to see you again soon at an upcoming edition of the EV Club, and we wish you the very best with your research. Take care, everyone.
Thank you, everyone.
Bye now.
Thank you.
Thank you, Jia Tong. Thank you.
Thank you.